we've already made it fairly clear that regulatory evolution is a critical, maybe the most important part of understanding changes in evolution and development. So what makes a butterfly look like a butterfly compared to a fruit fly? And when we look at it, most of the things that cause those big differences are changes in the regulation of very, very conserved developmental genes. So PITX1 is found widely across many organisms, and we talked about how in the fish there was this mutation in the area that regulates the hind limb, and so they no longer grow those big spines. And so that was a really nice study, and the reason that they identified that gene and then were able to identify the specific mutation was that they did a QTL study to find the gene and found that place with a really strong association between the phenotype and that region of the chromosome. Right there was PITX1. Boom, they knew that that was the gene. And then they could do more work to figure out what the exact change was. Now, there are sometimes changes in the genes themselves. Those amino acids will change, the genes will change, but those genes in many cases are remarkably well conserved. And after millions or hundreds of millions of years of evolution, when you see two genes that are incredibly conserved, that's an indication that there's been very, very strong purifying selection acting on that gene. Meaning that when mutations have occurred, because they have occurred, right? You can't just stop mutations. So mutations have been happening all the time. But when a mutation occurs, if it changes the amino acid sequence, it's almost always going to be a bad thing. And there's very little flexibility for this gene to evolve to take on new function. Now, quick review, and then we'll look at CREs. Why is there so little flexibility in these developmental genes? And we can basically categorize that under two major topics. Number one, most of these developmental genes are pleiotropic. They have 2, 3, 4, or as many as a dozen or more jobs that they have to perform in order for development and the organism to survive. So genes that are pleiotropic are more tightly constrained. There's going to be less chance for them to change and still have it be neutral or beneficial. So they stay very, very conserved. And then finally, developmental genes, particularly at the early phases of development, are vitally important. If anything changes, even minor changes, that can be a huge ripple effect and have a massive impact on the phenotype of that organism, and there's a very, very low chance that that's going to be anything beneficial. So the fact that they are so important in the way that the animal looks and the fact that they're mostly pleiotropic means the genes themselves very rarely change. And so this is why we can see incredible amounts of conservation, so that the eyeless gene from a mouse can be put into a fruit fly mutant that doesn't have a functional version of that gene, and it can basically just swap out. Okay, so let's talk more about cis-regulatory elements in detail. Paper 12 also goes into some detail about this. You should skim over it. I will sometime in the next couple of days put a little bit of a guideline for paper number 12, because there's, as in most papers that we read, there's tons and tons of details, and so I want to make it a little easier for you and just highlight the main points there. Okay, but here is our working hypothesis, or I should basically call this the modified hypothesis, once we begin to look at and evaluate all of these developmental genes and their regulatory changes. So changes in pleiotropic cis-regulatory elements give developmental genes the flexibility to be the major motors of evolutionary change. Now, they're not the only cause, but the major cause. And this still maintains stability necessary for the coherence of populations, so the integrity of populations, so they don't have so many changes and so many variabilities within that population that it becomes detrimental. And so it's really a great system for the ability to change and evolve, but yet still maintain integrity of the genetics underlying that population. And so it's useful, as we are studying all of this diversity of life on Earth, to think about organisms in a metaphorical way. And one of the metaphors that I like, that I think really helps me understand it, is to think of organisms like buildings. So all sorts of buildings around the world, whether it's the Great Pyramids, Greek temples, This is a famous cathedral, St. Basil's Cathedral in Moscow. This is a museum in, I want to say Barcelona. Anyway, it's an art museum. And although they look very, very different from one another, right? This is a wide range of architectural styles. They have some features that are identical, that are the same, right? So they need a solid foundation made out of brick, or I'm sorry, stone usually, or later concrete. So it's really, really critical that as we evaluate organisms, we look for those underlying themes, those things that they all have in common. What do they all do that's the same? And that actually helps us to classify major groups of organisms. And so animals have some early developmental mechanisms that are the same whether you are a snail or a segmented worm or a echinoderm or a vertebrate or any other animal. And then we can look at things that are specific to, say, chordates. And so these become critical markers for what makes animal group A different from animal group B. But there are going to be some basic foundational things that are the same for all of them. And then all of the details and all of the different genetic networks that are turned on and turned off in different locations create the vast differences that we might see if we just look at them superficially, like in a morphological point of view. Okay? So we're going to make some points here about regulatory evolution, some of which we've hinted at, maybe even done a little bit explicitly, but let's make sure you're clear about what each of these means. Regulatory evolution is one of the things that enables the pleiotropy of toolkit genes. So for instance, PIT-X1 has 4 main functions. It functions in the development of the thymus, the hind limb, the neural mast, and the olfactory pit. And the reason that this one gene can have all those functions is that it can be turned on differentially because of its diverse regulatory elements. And then there are PIT-X1 receptors, right? These regulatory elements that bind PIT-X1. They occur on all of the target genes for the development of these 4 areas. So you can see how those different regulatory elements allow for multiple functions for one gene. And so a gene that is a transcription factor, don't think of it as it is going to do the work itself. It's basically like a contractor of the house, right? The general contractor, doesn't do any of the building himself, but he goes out and he hires a framing crew, and he hires a electrician and a plumber and all of those things, and then gives them the plans and specific instructions. So that one individual can have massive impact on the way that the house looks by turning on, or in the metaphor, hiring different organizations to do his work for him. The same thing as transcription factors. They themselves don't do anything. They turn on genes that do things. Or really in the early stages, they turn on genes that turn on other genes that turn on other genes. And sometimes it's 4 or 5 or 6 steps removed before you finally get a gene that's actually doing something like cell proliferation or cell migration or other things. But it's doing something rather than just simply turning on another gene. So usually there are 5, 6, 7, 8 or more layers starting from the beginning before we start getting genes that are actually doing things. Okay, so this is an example with UBX, right? UBX can come on and make halteres in the flies because the regulatory elements that UBX product binds to are different in the fly than in the Lepidoptera. And so we don't make halteres, we just make fully formed hind wings, different in shape but very similar to the forewings. And then in the beetles, they have more of this ancestral wild type with some modifications. But that's because there are different downstream targets for UBX in these different organisms. Okay? So, number 2, regulatory evolution enables us to have developmental modularity. And what I mean by developmental modularity is we can start with a basic ground plan. So for crustaceans, for instance, ancestrally, every single segment was homonymous. Basically meaning that after the head, every segment looked about the same as the one before it and the one in front of it. They were all identical, much like modern centipedes. But today, of course, many crustaceans have vast differentiation of these different segments. So if we have a gene that is turned on in the the tail, the telson region, right? The telson is the very end, but in this tail region, that is not expressed in the cephalothorax, and that is binding to and turning on things that can initiate developmental patterns that are not available in the cephalothorax, right? And so this modularity then can be the ground plan, but then be worked on differently in different segments to yield very different segmental results. Some organisms still maintain the more ancestral form where they have modules, but there's been no differentiation that has occurred. And that works well for them. Oftentimes, these guys will have nearly as many, in the arthropods at least, nearly as many Hox genes. They just don't have as many specific functions because those differential functions haven't evolved in this group. But in crustaceans, the example we used previously, they're incredibly diverse. Okay, so we're going to actually be looking at diagrams. Now, this is one that I started with. I'm going to kind of outline how it works. And then I've got simplified versions of this. So in this diagram, the gold base of the arrow and the arrow over here represent the exons, the coding region of the gene. This area with all of the different globs attached to it is the promoter. So all of these quote-unquote globs represent the RNA polymerase complex, the RNA polymerase and the associated proteins that bind to it to help it to turn on and activate a gene. And then these little spheres represent different proteins that have bound to different regulatory elements. So I want to make sure you're clear in your head. The regulatory element is the actual piece of DNA, and typically they are 6 to 12, sometimes a little longer, but 6 to 12 base pairs that are very specific and will bind a regulatory, a protein, right, of the protein product of another gene. So the regulatory element will bind the protein. Okay, and we'll look at how many combine, and it's really a why, but we want to kind of introduce you to them and talk about some of the general trends that we see. But there's a wide range in what they can do. So the gray areas represent the rest of the DNA strand. Now notice regulatory elements are upstream of the promoter, and that's where most of them are found. However, sometimes they can be found in the introns, so in the non-coding regions of the gene sequence. They can be found there, and so there will be some there. Now, the name cis regulatory element implies that it's near the gene, so we're not going to have them far away on different chromosomes or on a very distant part of the same chromosome where we find the gene. That's just very, very uncommon, and there's not much evidence for that sort of thing. So really, we need to look near the gene, in the general neighborhood of the gene, for these regulatory elements. Okay, and what ends up happening is because of the multiple regulatory elements that control a gene and the fact that they might bind more than one protein means that we can have a massive and very diverse set of outcomes. So this gene can be turned off completely, it can be turned on a little bit, it can be turned on very, very rapidly or turned on slowly and ramped up. So depending on the combination of these that are bound, we can have different amounts and different timings for how this gene is transcribed and translated. Okay? Now, when a regulatory element changes, it can create something incredibly new, something that we've never seen before, or at least a modification. It can create something we've never seen before, or at least something in a new area that we've never seen it before. But it can also modify a structure that we already know about. So this is that example from the butterfly wings, Lepidoptera, where a change in the regulatory elements resulted in a hair-like structure turning into these plate-like scales. That was a critical, important step. And the gene itself didn't change. It's the same gene before and after with basically the exact same amino acid sequence. But how it's turned on and where it's turned on has led to this very different outcome. Same thing, we looked at the jaw example, how a change in Hox gene expression, so if the anterior boundary of the Hox genes is pushed back a little bit more, then suddenly these first couple gill arches are no longer under the control of those Hox genes, and other genes were then available to evolve, and as mutations occurred, they took on the patterning of those first two gill arches, and eventually those patterns, with many different mutations and control elements that became part of this. But those patterns led to the origin of the vertebrate jaw. Both of these are novel features. Now, we understand their origin, where they came from, and if we don't understand all the steps along the way, we do know about some of them and have traced some of them to their genetic origins. Okay, so we are going to look at 4 characteristics of these regulatory elements, and these are generalizations from what we have learned looking at all regulatory elements in animals. Okay, and plants, really, this is really a eukaryotic thing. Okay, but most of the work that we have done is in animals, particularly in the arthropods. Okay, so most cis-regulatory elements are regulated by anywhere between 4 and 6 transcription factors. So that's kind of a general rule. But realize that's general rule. Some of them can have, a single regulatory element might bind more than 6. The higher you get, the more and more rare it is. And there are some regulatory elements that are incredibly specific that will only bind a single transcription factor. So here I've got 4 of them in our little diagram, right? So this is our base diagram, a simplification of the one we saw earlier, with the P being the promoter. And here we have 3 regulatory elements, okay? So this single regulatory element here labeled number 1 might bind 4 different proteins, and I've color-coded them, right? So if the red one is there at a certain abundance, it will be the main one that binds, and whatever impact that red protein has will be what is holding sway for that gene in that tissue. But if we ramp up expression of the blue protein, maybe it will then competitively bind, push out red proteins, and then it maybe have a different impact. So there's going to be competitive binding because only a single protein at a time can be bound to this regulatory element. Okay. The spatial relationship of binding sites is important. If they're really close to the promoter or really far away, that can have a difference. So not only the regulatory element itself and how many proteins it can bind to, but where it is located can play a role in its evolution also. Okay, so for many flexibilities, for many regulatory elements, there is some flexibility. So if you move it 10 or 20 base pairs one way or the other, it may not make a huge difference. But if we move it 100, and certainly if we move it from a intron to upstream in the non-coding region near the gene, that's going to have a dramatic impact. And it might still have some function, but it's not going to be the same as it was prior to its location change. Now, if a regulatory element is unbound, meaning there's no protein stuck to it, the ground plan state for that gene is to be turned off. So most genes are just kind of off. That's the default setting, if you will. And so we need things that will come in to turn them on, to activate them. Now, some regulatory elements have an additional repressive effect. So it's turned off if nothing is bound, and then if maybe we'll say regulatory element number 3 has a repression effect. And so if the protein that binds to it suddenly becomes available, that gene continues to be turned off and may stay turned off despite having other genes binding to it that are trying to turn it on. There's kind of this antagonistic impact or give and take between different regulatory elements. And so this gives us many different ways and mechanisms by which we can control the expression of these target genes. So the spatial boundaries of gene expression, and we could say temporal boundaries also. So basically where and when genes are expressed during development, these are set by this positive and negative impact. So in this example, I've got a purple protein representing a repressive effect. So this is binding to that regulatory element and repressing expression of that gene. Whereas another one might turn on, but it can't bind. So a little X represents the fact it can't bind to it because it has the same binding site as this purple one. And so we might need to ramp up this blue protein at much higher levels than we normally would so it can outcompete and bind here. Whereas if the purple protein was not present in these cells, it would be able to bind very easily, and that gene could be expressed and turn on very, very rapidly. Okay, number 5. Terminal differentiation, so the very last in a series of a hierarchy of genes being expressed. This terminal one is usually much, much more specific than the early ones. So the early ones usually have more regulatory elements and can be turned on by many different things. But as we go farther and farther down that pathway, the more specific it gets. And so by the very end, there might be, and this is not pictured here, they all just have kind of the general 1, 2, 3 regulatory elements, but we might have fewer regulatory elements and regulatory elements that can only bind one very specific product to be able to turn on that gene. But these are the ones that are not going to have as much of an impact on the phenotype because they are later in the developmental process, right? So if we change a maternal gene, one of these genes that's critical for very, very early patterning of the embryo, we end up with a massive genetic and then phenotypic difference in that organism. Whereas if we turn off a gene that's 12, 13, 14 or more steps down the line, we're going to have a much less of an impact. And then finally, number 6, all of these concepts that we've talked about mean that there are incredible variety and diversity and incredible flexibility in the way that we can turn on and turn off genes. So it gives us kind of a, I don't know what the word is, a very complicated starting place. The raw material for building organisms after hundreds of millions of years of evolution have duplicated genes and built up more regulatory elements and changed and evolved them. It's made this incredibly complicated genetic control system for building an organism. And so we can change it and have new species or new variation in the species and still maintain the integrity. So an analogy that helps me, it's kind of like, I just pulled this out, this is a little bit dated, we've got an iPod on there, but it's kind of like these complex soundboards. If you are a DJ or a music producer of some sort, right, you can have a massive impact with the same basic starting point. So if you had somebody play some music for you or sing a song, there are all sorts of different things you could do to that. You could add sounds to that, or even if you weren't adding anything, if you were just manipulating the sound that was already there, right, you could add auto-tune to it, or you could increase the pitch, or you could, do all of these different things that would make the outcome very, very different. So this is a little bit like organisms with the same basic set of genes with only a few minor differences, right? You can take a embryo that's going to normally grow into a chimpanzee, and with, I don't want to minimize the number of changes because there are many. Most of them are regulatory though, right? We didn't change the genes a whole lot. We mainly changed how those genes are interacting with one another and when and where they are expressed. And so the difference between a chimpanzee and a human, or a fruit fly and a housefly, or any other two examples, right, really boil down to these complicated control mechanisms, the cis regulatory elements that are turning on and turning off genes in the right amounts at the right time to lead to very different outcomes for the developing embryo.